Wait, hold on, is it working? Yeah, okay.
So, we take Golgi apparatus and ligands for Dynabec and oligo brand.
Yeah.
So, what is the structure made of? And Golgi apparatus is made of like a stacks of membranes, which are called cisternae, and they help proteins to travel within the Golgi complex, like through the Golgi complex. And it has 5 to 8 cisternae each stack, so like each stack there's like 5 or to 8 of those. But proteins can have like up to 60 in each stack. And Golgi enzymes are found inside of each cisternae, and later we're gonna explain that in each cisternae, like there's like different like locations, and each have like different enzymes where proteins are modified. And an individual stack of cisternae are called dicotysomes.
Dicotysom, maybe?
Dicotysomes, and it comes from the Greek word dicoton, net, soma, body. And mammalian cells can have 40 to 100 stacks. And this is a picture of it. So these are like the cisternas, and so there's like a lot of stacks of those. And like later the proteins will go and they'll like go through the Golgi apparatus like that. And so these are the 4 functional regions. The cis-Golgi network is where, so the Golgi apparatus is near the rough ER, and proteins are made in the rough ER. So those proteins are like moved to the Golgi apparatus, and this is the first place where the proteins go through, and they go.
Isn't it this one?
No. Oh, this one, trans, and then endomedial, and they go out in vesicles, in little vesicles. And those vesicles can stay inside the cell or can be exported out of the cell. And each region contains different enzymes. So this is a picture, and the plasmid reticulum, cis, medial, trans, and select the proteins going through each one. Another picture of it.
And how was it discovered? It was discovered during the investigation of the nervous system using the staining technique. And they found it out with a microscope, and it was believed to be an optical illusion. And in the 20th century, discoveries disproved that. And when and who discovered it? It was one of the first organelles that was ever observed due to large size. And it was discovered by an Italian physician named Camillo Golgi. And
so basically this whole Golgi apparatus organelle looks like a stash of pancakes. Yeah, if you have to compare it to a different organelle, you could sort of compare its shape. Yeah, it's viewed to the smooth endoplasmic reticulum. I like the pancakes a lot actually. And so yeah, so what does this organelle do? It has 3 main things. It focuses on the proteins, lipids, and lysosomes. Well, for the proteins, as Minchi just explained, the proteins come from the rough endoplasmic reticulum. They come, they enter the Golgi apparatus, they go through, and the Golgi apparatus it modifies the proteins. It adds carbohydrates and phosphates to it, so other so the cells and enzymes recognize it and know how to build with it. And it's also for intercellular recognition. Here we are. And then for the lipids, it's basically just for transport, and it produces lysosomes. The lysosomes are the ones which break down unused or cells, parts within the cell which are not going to be used anymore, and those are recycled.
And the proteins with the carbohydrates we saw in the cell membrane are made in the Golgi apparatus. So there's like a genotyper. And this like as you can see, like it goes like vesicles and it goes through the cell membrane and it comes back, and there's like a cycle kind of as the cellular respiration with that. And so it goes like proteins, it comes back as proteins and goes like it goes through all the processes, goes back to the Golgi apparatus, and like the cycle never
Yeah,
stops.
just like Benedict explained this whole cycle thing. Golgi apparatus has it as well.
And these are like some of the proteins and how they're modified from each stage of the Golgi apparatus.
Yeah, so we find this Golgi apparatus in animal cells as well as plant cells, and yeah, that's it.
Okay, thank you.
Bravo.